and I appreciate each of you being here tonight, and I hope that I'm able to communicate something that has really stirred my professional and personal interests. I am the medical director for Manna Relief Ministries, and our mission is to get dietary supplements, the technology that I've been part of a team that worked on nearly 20 years, to children, to assist in getting it to children whose parents could never afford these products. Because we have found that children get the greatest benefit of anyone, but their parents, and their grandparents, and others do too, fortunately. On the screen is a cover for a syllabus to the 3rd National New Initiatives in the Prevention and Intervention of Fetal Alcohol Syndrome and Fetal Alcohol Effects for Aboriginal Peoples of Canada. And if I were to give you a title for what I'm attempting to do tonight, it would be A New Dimension, : autogenous stem cells, adult stem cells from your own bone marrow, are supported in their production and function by glyconutrients, sugars that are missing in the modern food chain, which is what got this story started nearly 20 years ago. The reason I was invited to go to British Columbia, Victoria, to present this is that they had 7 children on reservations in Canada that had been placed with foster parents and adoptive parents taken away from their alcoholic mothers with terrible fetal alcohol syndrome. I began to learn more about this, and the reason I was invited is they said that these children had added to their diet these dietary supplements, and they had major improvements in their behavior and learning capacity and functioning capacity. Once we were told about this, we tried to get an assessment or evaluation about just what had happened. And we found on the internet a questionnaire that has 36 questions that are asked of the person that takes care of and knows a lot about the child that is school age, usually administered above 5 years of age. And they ask 36 questions. Here are the first 3. Does this child overreact? Yes or no? Chats with no content? This child, no. Talks of unusual and bizarre topics? No. This is usually applied one time, and you predict the future of this child for the rest of their life. They said they improved, so we modified this, and if they said a yes, beneath each one of them, we had on a 0 to 10 scale, from normal to very abnormal, before the dietary supplements or micronutrients were added to the diet. How bad was it? And that's a square. Very abnormal. Then they were asked, now that it had been a minimum of 1 year that they had taken these dietary supplements, how would you rate them on this problem now? And this is the type of thing they got. Now, how such improvements could happen in fetal alcohol syndrome children? We can say through optimized nutrition that supports better cell synthesis of life-conducting molecules. And these molecules are glycoproteins, being glyco from the Greek, meaning sweet or sugar, hooked to chains of amino acids, and sugars hooked to fatty substances. And they are the structure-function molecules of our body. Our cells make these and synthesize these from what we eat and what is in our diet. And then now what I refer to as the new dimension, that these improvement in nutrition also supports generation of stem cells from the child's own bone marrow that can differentiate to any damaged or destroyed cell or cell programmed for renewal or replacement, which is apoptosis. As we age, you've probably heard the figure I have that we have a new body about every 7 to 8 years until we start getting older, and the programmed destruction of old and aged and damaged cells slows down, and their replacement begins to slow down, and that's part of becoming feeble. Well, this goes on all through our life, from very small numbers of stem cells from our own bone marrow that generate in and migrate into the tissues and renew and regenerate our body. Now, in regard to the first thing about nutrition, I have used this as an example that if you understand how General Motors, Chrysler, Ford makes vehicles, you can understand the importance of nutrition in our daily life. That parts are brought into the assembly plant where there is design and engineering plans or a blueprint, and if you have all the parts brought in by rail or truck, you put them together right, you get out a completed product that looks right and works right. If you've got that, you don't need a college course in molecular biology, genetics, or biochemistry. But why I tell you about this is that each cell in our body, though under a microscope, it must be enlarged 500-fold just to see this much detail, that under the microscope, the blueprints of life are in the center in the nucleus, where the chromosomes you inherit from your mother and father are present, which are the coding for the synthesis and how you handle nutrients that we put in our mouth and begin to chew and get them smaller, because, as I say, each cell is smaller than the point of a pin. And yet, what goes on in that cell is more complex, more involved, more detailed than in a huge assembly line plant that makes vehicles. Well, we put nutrients in our mouth, we call food, and through the digestive process, it brings it down to molecular size. Enzymes chop large molecules down to amino acids, sugars, fats, vitamins, and minerals, absorbed in the blood and pumped by the heart in the serum phase around to supply parts to the cell. And that's why nutrition is so important. We had been working for a number of years as a team trying to figure out why human beings had been using aloe vera leaf gel, for over 5,000 years, that it can be documented. And it was found that a white powder could be extracted from the gel, and that it was chains of mannose sugars. By tissue culture in animals and then in humans, we were able to show that this is why human beings have been using aloe vera for over 5,000 years. Mannose sugars. When we announced this at meetings, no one professional wanted to be seen talking to us afterwards, because medicine and science had long ago decided that sugars are burned for energy, and that hormones, DNA in our chromosomes, proteins were the important molecules of life, and that sugars were mainly firewood for energy. But in this paper, it showed that in the second step here, 9 molecules of mannose sugar are required to be added to protein chains coming out of the ribosome, and that this is a very important chassis or domain, because this is where the charge and configuration is that keeps our cells connected to each other, that should stay where they are. We don't want our skin, our lungs, our liver wandering around. When they do that, that's usually cancer. And interestingly enough, there is a gene defect in this. It's called epidermal isobulosa, and we have a number of children that by supplying more of this, they quit waking up in the morning and leaving hunks of skin in bed, and they have all the problems of infections of people with severe burns. But this is also a domain for cell-to-cell communication and instructions. And as this progresses along the assembly line in the Golgi, other sugars are added or substituted to these 3 chains for cell-to-cell instructions and coding, so that because we have other cells that don't stay at home, white cells in our body that defend our body, but they're spread all through their body. You're aware that with an abscess, pneumonia, kidney infection, you can see thousands of them. Your urine become cloudy, you cough up green or discolored sputum, millions of them in your sputum, or in an abscess, purulent material can be expressed. Millions of these white cells. Did you ever wonder how they knew to go there? Well, a message was written to them on these 3 chains. It was finished off in the Golgi, and more detail on this is shown in this, that the original sugar from the mannose from the aloe forms the chains, and then as it goes into the Golgi, other sugars are added and changed to allow cells to send the word to white cells, I have found a virus, a yeast, or a bacteria at some place in the body. Come here, destroy it. Ooh, there's a mess here left over. Bring in other types of white cells called macrophages and clean it up. Oh, there's a defect here. Written on this chain here is we need to proliferate fibroblasts to make a scar. And you're still oozing or leaking if it's skin or in the GI tract. We need to have epithelial cells to grow and cover it up again. All of those wonderful instructions are written on this. And so we went to nature once we understood the importance of mannose and found these other sugars. We have plenty of glucose in the modern diet. If you drink milk, galactose. But these other sugars are low because we plowed up our gardens, chop down our orchards, and process our foods. One of the reasons we found out mannose was so important was not only its role at this important step, but when we added it back, we had parents, patients, doctors, nurses saying, look what happened, it's a miracle. I'm here tonight to tell you it is no miracle. That this is very vital and important. And the reason it's so important is that even though we raise it by the tons in grain fields and rice paddies, but when you make white rice and white enriched flour by the processing, you strip the mannose out so that when you add it back, people say it's a miracle. Well, you can kind of appreciate, I believe here, the importance of this micronutrient that we call now a glyconutrient, a sugar that is vitally important. As we begin to reflect over the fetal alcohol syndrome children and their improvement, and I was using the analogy of the automobile assembly plant, this did not seem adequate to define what was happening in fetal alcohol syndrome children and cerebral palsy children. That as medical director for 8 years for a marketing company for this product, that I would hear time and time again from parents. My child was in a big medical center with severe cerebral palsy. Year and a half, 2 on these micronutrients. They've dismissed them, said they can't find anything wrong. They made the wrong diagnosis to start with, that this doesn't happen in cerebral palsy. Or leukodystrophy, where there's a gene defect and the brain doesn't properly develop. Or in autistic children, where we heard many reports of benefit. Or in Down syndrome children, where children came out of special education and into regular classes. This did not seem enough to explain how adults with Alzheimer's, Parkinson's disease, Huntington's chorea, oligonuclear degeneration, multiple sclerosis, and amyotrophic lateral sclerosis, in other words, more commonly known as Lou Gehrig's disease, it had benefits. What in the world is going on? Because if you raise the hood of a car and the motor's missing, just talking about a few missing parts doesn't seem adequate. So as we had begun to try to assess what was happening in the fetal alcohol syndrome, these other conditions I'm telling you about that are considered to be permanent. But I think possibly my prayers were answered when on February the 19th, 2003, in the Journal of the American Medical Association, here is a short review of an article they thought was so important that was published in the Proceedings of the National Academy of Science. Behind this story, I want to tell you, it's very briefly here, that in bone marrow transplant research with animals, Dr. Eva Mezzi states that we've known that stem cells from the donor mouse injected into a recipient mouse have crossed the blood-brain barrier and been found to be neurons in the recipient mouse. And she said, we thought probably that was fine for mouse kind. but surely not in human beings. But she had a friend at Johns Hopkins Medical School, Dr. Barbara Crane, who's a pathologist. And this tells you here that they had 4 females, 2 teenagers, 2 in their 20s with leukemia. They had all the treatment prescribed for leukemia, the best they had to offer, including bone marrow transplants, only from male donors. And they died. They came to autopsy and they got their brains. And what they're showing here is what they found in all 4 females when they took sections of their brain. And this is what a neuron looks like, the body with the axon, specially stained under fluorescent microscopy with 1,000 magnification. In their brains, they found significant numbers of neurons that had a little red glowing dot because they made an antibody very specific for the Y chromosome, which is only present in male cells. Women are XX. And these 4 women at Johns Hopkins that had been treated for leukemia, that had male bone marrow transplants, all had neurons that came from stem cells from the male donors, adult stem cells. But that's a pretty good trick because the brain is protected. There's a blood-brain barrier. And these cells, as Dr. Mezzi said, like from the tissues, there's a signal. Come here, migrate in, and become something that is needed to replace something damaged. And here you see it in the brain, and this is irrefutable proof that adult stem cells from a donor have gone into the brain. Well, when I read this, I was impressed because I was taught that when you're born, you have billions of neurons in your brain, and you start losing them. Those people have a very difficult or complicated delivery and get cerebral palsy. That's what happened. You lost a whole bunch. And you never get them again. As people get older and we say grandpa or grandma's getting feeble and forgetful, you've just worn out a bunch of them and you never get any new ones. Well, it's now known that that is not true, and this is part of the proof of it because this process goes on from your own stem cells as part of the word I told you before, programmed cell death and renewal apoptosis. But that's not the real thing that is the cause of me saying this is a new dimension. What I'm showing you is a blood smear from back in the 80s that left a puzzle that was never answered until this paper was published. We found out that in toxicology studies in dogs in trying to make the white powder from the aloe, the polymannose, a drug, that when given to dogs in their peripheral blood within a week, there were huge cells that were usually not present in their smears. We eventually found out that in humans with AIDS, with cancer, with the autoimmune ulcerative colitis, and some controls, that a week after starting on the active ingredient from the aloe plant, that huge cells were seen in the peripheral blood. And for orientation, this is what a normal red cell looks like, about 6 microns in diameter, that carries oxygen to the cells. These are little platelets, 1 to 2 microns in diameter, that are involved in keeping the blood from flowing too readily and clots. These are lymphocytes with a nucleus of, again, about 5 to 6 micron in diameter, and these are the type size cells you usually see. But here are huge cells that the nucleus alone with the chromosomes is larger than the entire lymphocyte. And the diameter is 10 to 15 times larger. And when the genes are turned on here and they start to make glycoproteins or glycolipids, as we said on that assembly line, this is what the cell looks like. And this is production on the assembly line in these activated big cells that for over a year we called G cells. Because when I first saw them under the side, I exclaimed, G, brought over the senior technologists. None of them had ever seen anything like it either. And so we just called them G cells for a year. Then we got our hands on a research flow cytometer. At that time, it was a relatively new and primitive instrument. And I don't want to spend too much time on the details of it, but when you talk about someone having a white cell count, maybe anywhere from 5 to 10,000 or elevated, in this instrument, every one of these is one white cell that has been pulled through a little orifice and counted electronically, and then they shine light on it, and the complexity of them and size of a shadow called light scattering, complexity goes this way and size goes this way, and this is before taking the aloe plant. And you'll notice where the red area is, there's nothing up here where big cells should be if they were there. After 1 week of taking the active ingredient from here are the big cells. And with the proper antibody, they were found to be LEUM3 monocyte macrophages, which are cells that have major potential and are possibly a surrogate marker, because you can't get more of these cells unless stem cells have differentiated along the line to become them. We didn't know what this meant, but we knew that it could support better defense, repair, and healing in the body. And for 15 years, it lay dormant until we saw the paper from the JAMA, Journal of American Medical Association. It dawned on us that this indicates and is the basis for how those fetal alcohol syndrome children went from an IQ of 50 to 100. Why the Alzheimer patients, the Parkinson patients, the cerebral palsy patients, why they improve, because these cells migrate into the tissue and can differentiate, are the family of cells that do it, stem cells, where they come from. The subject of stem cells is major, and of course the big issue in this country is embryonal stem cells versus adult stem cells. I want you to ignore the upper half of this, and the bottom half of it is what is known as the blastula stage of a human's development. It is a ball of cells after the sperm hits the egg, and it's usually in the fallopian tube, that there's a rapid growth and there's a single layer of cells, and it's means berry. But the important thing about this is every cell here has the capacity to become anything in the human body. Early microscopists and histologists and embryologists in this stage would take micro dissection cells here, move them over here, move them up here, and you'd get a normal developing animal if you did this at the blastula stage. And this is what we have over 400,000 of, frozen in liquid nitrogen, in North America. And there are scientists that want to take these and put an enzyme on them that'll break them apart, put them in media and try to get them to grow, and then bore a hole, for example, in the skull, and with proper technique, insert a needle down into where there's damage, say from Parkinson's disease, and inject these cells in hopes that they would repair and regenerate and stop the Parkinson's disease. The next thing you need to know is that if you'll look now at the entire blastula, the next stage is called gastrula, with a G. And at this stage, a disc forms on the blastula, and you have cells inside here, cells in the middle, and cells on the top. In Latin, that means ectoderm, mesoderm in the middle, and endoderm. If you move these cells, you end up with brain tissue sitting in the muscle or in the liver, or liver cells in the muscle and in the brain. You no longer have total potential of stem cells to become anything needed or damaged in the body. And that's why there is such interest in this type of thing. Now, I want to try to simplify what I've been trying to tell you, that the total development potential for targeted cell replacement runs along exactly the same path that happens in the human development. For example, I was telling you in the blastula, which are 100 to 200 at the most, total potential cells. At the next stage, the gastrula with endoderm, mesoderm, and ectoderm, that there are fluorescent antibodies that now are available for the flow cytometer that can tell you what kind of cells you have in the peripheral blood. For example, we have CD, that means cell domain on the membrane where the antibody reacts 34, and that's an adult uncommitted stem cell, which is equivalent to the 100 to 200 total potential cells in the blastula. These cells from our bone marrow can migrate and become anything in our body that's damaged. If the cells differentiate further and wander into the bone marrow, the fluorescent antibody BB9 goes to the bone marrow, and from this, other elements in the bone marrow, whether they would be red cells or megakaryocytes for platelets or monocyte macrophages, and those that are going to the ectoderm or the brain, neural plate and tube, or CD90. These antibodies here did not exist back in the 80s when we first did this, because I told you that those cells tagged with the LUM3 was the antibody that was available, and now it's called CD14 as this has developed. And so that's just why we were able to come to this conclusion, and right now, with the support of Kathy and Tom Keller Foundation and the Agape Foundation, there's money left there. We've had some funds to start redoing all this and using these new antibodies to answer the question that will prove unequivocally that this is how, through nutrition, we're seeing benefits and regeneration in the brain and even in children with type 1 diabetes. It's been shown that in the endoderm, that cells migrate into the pancreas, become beta cells that make insulin, and we now have type 1 diabetics that can't take insulin because through this process they have replaced the insulin producing cells in their pancreas. That's the power of what we're trying to get across to you. Now, and another issue, there's been an explosion in stem cell research. And this is just an example that explains a little girl that we had in the Cleveland, Ohio area, Emily, who had Duchenne's muscular dystrophy so bad that she went from crawling to the wheelchair. I forgot to put in here the picture of her on a parallel bar in gymnastics. She is now excelling in gymnastics out of the wheelchair from taking these dietary supplements. I've just got through showing you how this is possible, but they've gone a step further, and this is from the August issue last year of JAMA. This is the leg muscle of a mouse that with a special technique, they knocked out the gene that essentially gave the mouse Duchenne's muscular dystrophy, and its leg was paralyzed. With stem cells injected into the mouse from a donor, they migrated in the muscle, and now the mouse no longer has a paralyzed hip muscle and has regenerated from stem cells, which undoubtedly is how the children we have had with muscular dystrophy have been able to throw away their crutches and get out of their wheelchairs if started young enough. This is an incredible thing about stem cells, that they have found that there are mothers that they've studied that have boy babies. And boy babies' stem cells, you can imagine the baby in the uterus growing rapidly, is a mass of uncommitted young stem cells which would tag with the CD34 uncommitted like the blastula. And that what they have shown here is that from the boy baby stem cells have crossed the placenta into the mother. And they have been found in the liver cells, in the epithelial cells, and in leukocytes, or white cells, of the mother. And the way they did this, and this is another one that had a boy baby. This was a needle biopsy of the mother's thyroid gland. In the mother's thyroid gland, there were cells that you can see here. Everywhere you see a green dot, it's just like the same as the antibody that was in the brain, but this is a Y chromosome and this is an X chromosome. This is a Y chromosome and an X chromosome. Stem cells from the boy baby have migrated into the mother's thyroid gland, and this is absolute proof of it. Here is even further proof. This is in a mother that had a boy baby, and this is a card of liver tissue, and this is a blood vessel between two cards of liver tissue, and in this blood vessel is a stem cell that has the Y chromosome, this little dot right here, and this is the X chromosome right here, red, but you see sort of a cloud of reddish brown. This is the CD434 stem cell, showing that stem cells from the baby are in the liver card, wandering in, potential to replace the liver cells that I showed you like were going on in the thyroid gland. And this is an example from one of the mothers with a boy baby. The liver cell, and see the little green right there, in the liver cell of the mother is the Y chromosome. Now in this issue there's an editorial, and they say, in showing this, this may explain why women have more autoantibody disease like lupus, rheumatoid arthritis, that they have so many cells that are foreign that they're reacting against them. That's the downside theory. The upside theory we can smile about. They say this may be why women live 8 to 10 years longer in males. Every time they're pregnant, they essentially get a flood of fetal stem cells that regenerate tissues in their body. And of course the answer has yet to be known. But I don't know whether that fires up your imagination and possibilities, but that's the nature and the power of the controversy that's going on in this country. Now, here is another recent paper, and this is from this year, about 2 weeks ago. Endometrial cells. If you'll stop and think, these are rapidly proliferating cells. Every month, if you're not pregnant, you build up a layer and then it sloughs off and you regenerate another layer. You can sample these cells with an aspiration device and look at these cells, and what they showed that the stem cells, these were a paper on women that had leukemia, and they did whole body radiation to destroy their bone marrow, and then added chemotherapy in large doses to essentially sterilize the bone marrow to try to destroy all the malignant leukemic cells. Then they gave a bone marrow transplant from another person, which would not be her cells. They show that this is the controls, and these are not using fluorescent antibodies, but are using other types that give you a dye. And any cells that stain the bluish purple color are the lady's own cells and glands and stroma in the endometrium. If the cells come from the donor bone marrow stem cells that migrate into the uterus and differentiate to make glands and stroma and blood vessels, they'll stain brown. Well, here it is. These 1, 2, 3, 4, 5 glands were from her own cells after all of this insult. These brown ones came and were endometrium from stem cells from the bone marrow donor after they had sterilized her bone marrow. This one was unique in that she was about 50-50 because she had not only whole body radiation, but she had 2 different chemotherapies in high levels to destroy as much bone marrow as possible, and they had to give her more stem cells, and so it's a dose basis. These over here and many of these were only about 2 to 5% of the cells were from the donor. This lady had 50%, so there was a dose response by the number of stem cells she was given. And these stem cells don't just look like it. This is showing on the end that the cilia on the, see the brown ones are from the donor. These are from her cells right there, her own stem cells from her own bone marrow. These stem cells migrated in and differentiated to make glandular cells, and they even have cilia on the surface. So they differentiate to a major degree. This goes even further to show that in the endometrium there are white cells in the blood cells and in the stroma. See, these are not glandular cells, but the blue staining are stromal cells that support the glands that are the mother's own cells and chromosome types. Here's another one. These that are brown are stromal cells from the stem cells of the donor, and then in a blood vessel here are even polymorphonuclear leukocytes that by their staining, they are not her chromosome source, but from the stem cells of the donor. This is a paper that gave an answer that I'd been looking for 2 years, ever since we saw the brain cells. And if you'll stop and think, we've shown you stem cells from donors have gone to the brain, the thyroid, to the liver stroma, the endometrium, endometrial stroma, and even white cells. You should be getting the idea pretty quick that adult stem cells can migrate into the tissues and replace any cell that needs to be replaced in one human being. Because what I showed you in that little cloud of dots is in 1 microliter of blood. It takes 1,000 microliters of blood to be 1 cc, or 1 cubic centimeter, about 2.5 per teaspoon of blood. A 175-pound man has about 3,000 cc's of blood. In 1 week, one human being has more stem cells in circulation than all the stem cells of North America. In the peripheral blood, in this paper, they call large mononuclear cells endothelial progenitor cells. And they're derived from the monocyte macrophage, and they even secrete factors that cause blood vessels to grow. But in this paper, it shows that these cells, right here, CD14, which is the new name for LUM3, that group of cells that I showed you back in the 80s, this is the name of the antibody. It's the same antibody that in the peripheral blood, these big mononuclear cells, and they went even further with others, and this CD11 is a subgroup of these that have differentiated and been stimulated, and they migrate into ischemic tissues anywhere there's a bruise or ischemic damage, like in a heart attack or in the brain where you have brain damage and you have blood in the tissues. These cells migrate to those areas. And it shows you here the CD34 lymphocytes do not cross-react very many, but this is what they're coming from the person's own bone marrow, because these patients didn't get transplanted bone marrow. These were their own. One thing left is the heart. And we've had a number of patients over the last 11 years that were told they were going to have a heart transplant if they were going to live. And they have gotten on these dietary supplements, and they have serial echocardiogram showing that the left ventricular, or the big pump that has to pump blood around in the heart, have turned around, got stronger, and they didn't have to have a heart transplant. Well, we wondered, how in the world is that possible? Well, these were 4 women that had leukemia, and they were treated and they eventually died. And I'm going to show you what they found in their hearts. 4 women that had leukemia, and they had bone marrow transplants. This is a normal section of heart of each one of them. And the reason we show this, an example of all 4, is there's no inflammation, no scarring, just healthy heart tissue, the type of thing a pathologist usually looks at. These are special stains that show where the nucleus and the chromosomes are, and the red stain tells you that these are muscle fibers of the type that are in the heart. And then we look at this nucleus here, and here again is the Y chromosome from the stem cells of the male donors that have migrated into their heart and have replaced aging and damaged heart cells. This shows you that not only the heart cell has the Y chromosome, but even the stroma that hooks the white cells together have Y chromosomes from the male donor. And this is another example of many of them in clusters where they think that there was scar and they had many of the stem cells from the male donor. Then in the muscle, skeletal muscle, another tissue that shows the striations, which are different from cardiac muscle. Here is the Y chromosome in these women's skeletal muscle and also in their liver. You can see several of them here, the Y chromosome in these patients. Are you getting the picture of how important these cells are and what their capacity are? A new dimension for health and healing in the human body. Now, one of the best examples to show the power of this is one of the children that we've come through in Man Relief Ministries, and I introduce you to Cole. Cole's mother, very emotionally, and it was gut-wrenching for me, told how when Cole was 18 months old, she and her husband bought a new home and did not know it was defective, much less that the smoke detectors did not work. The house caught on fire. She described how Cole's older brother of 6 years of age died in her arms before the ambulance could get there from smoke inhalation damage. Cole was unconscious, and he was unconscious for a number of years. He was so unconscious they didn't even have to do a tracheostomy on him. He had no reflexes. The only evidence of life in Cole was a heartbeat, and he breathed, and he was fed by a feeding tube. In the year 2000, when he was approximately 3 years old, they did a 19-lead EEG for brain activity while he was in deep coma. Someone heard my lecture on how this, and I have simplified it for people, saying you don't need to know all the technology. You just try it and it works. The science is to open your mouth and swallow. Why swallow? It contains what your body was designed for and has been provided in food and nature. Wonderful things happen. You don't have to know a lot of details. It's good to know this kind of science has been done. But someone went to Cole's mother and suggested that the micronutrients be added to his feeding tube. In a month, he moved his right hand, the first evidence of any activity, for over 3 years. He continued to develop, and at 6 months he had come so long that they decided to repeat his brainwave activity. And here he is as he's come out of coma and moving his hands and lungs and legs. I'm here to tell you that those cells that look like they migrate into the brain or in the place that stem cells should go and look like also work, and this is the proof that they work and have worked in many different conditions. Here is Cole when he was in deep coma. Here he is looking at me, focusing on his left eye. And this is a little girl, Chloe, who Cole's mother told Chloe's mother, who had been in an auto accident, had been in coma for many months. And within less than a week, she came out of coma and is slowly beginning to develop. This is just proof that I went by to see him. When Cole went into coma, he wasn't talking. And he had made no utterances. And his mother made some comment. I said, you ought to double in his feeding tube the amount of the complete glyconutrient formula. In 2 weeks, over the phone, she called crying that when he sees me comes in the room now, he looks at me and he goes, ooh. Now, if you could see his mother, you could say, oh, that's an appropriate sound to make. But they brought in the speech specialist, And I was told by phone that he started kindergarten last September. I introduce you to another child that have had benefits, and I like to emphasize that this power is more potent in children, and the younger you can get them started, the better. In the entire 19 years that I've been involved with this is the one individual that has benefited the most from this technology. That doesn't treat, that doesn't cure. All it does is improve nutrition, parts, if you will, that are required by the programs of life that you inherit from your mother and father and genes that are the instructions for normal structure function. It is not normal to be born weighing just a little over 2 pounds, 3 months premature. To a mother that abused chain-smoked cigarettes, which causes prematurity and small babies, who abused alcohol and drugs, and at the medical school in Colorado, Sarah was diagnosed in the ultra-premature nursery room as having fetal alcohol syndrome severe, cerebral palsy, sensory integrative defect, and immature heart development. She had septal and cushion defects in her heart. They did not recommend that she be adopted because she was going to need about 3 heart operations, which she may not live through. And then you would probably have a baby with an IQ of less than 50. And if you're not familiar with fetal alcohol syndrome, this is where I learned. I thought you had to be a fall down, staggering drunk when you're pregnant. That's not true. All it takes is one good night, one good weekend, maybe 2 or 3 stiff drinks at the wrong time. And the damage to the brain can be what has been before this, permanent and complete for a lifetime until this. Now, when they're babies, you can't have a 36 question thing, so they measure the skull and x-rays, and her eyes were too far apart. The brow above the nose is atrophic. One eye can virtually look across and see the other. The nose is turned up kind of like Porky Pig. There's a very thin upper lip, and of course she had the heart problem. As she began to toddle, this is the face of fetal alcohol syndrome, and there are very critical measurements that say this is a severe fetal alcohol syndrome. She probably will never learn, train, live independently. I was called and asked, would these substances for the diet help Sarah? And I told them the truth. I don't know. I would like to be able to tell you that every person that takes them gets a miracle that they're looking for. That is not true. It's not 100%. As I frequently say, the only thing I've found is 100% is death, taxes, and scoundrels will run for public office and get elected. That's 100%. I told them how they should put the products in their formula, and she said, well, she's not getting formula. She's getting something in a tube down in her stomach. And I said, well, there's not much baby there. The skin's premature and all. We have this in topicals that are gels and creams. Do you get to hold her? And they said, twice a day for 30 minutes and rock her. And I said, well, the whole time you're rocking her, apply this to her skin from her hair to her between her toes. And they did. And then when they could put the formula, they put it in her. And I didn't hear from her until she was 5.5 years old. And this is little Sarah. Now, this was sent to me with the statement that in evaluating her at the medical school, the developmental pediatricians are puzzled. They find no evidence of fetal alcohol syndrome or her cerebral palsy. That now her eyes are the right distance apart, the tissues filled in here, her nose has come down, her ears are in the right place, her skull x-ray is normal. Her IQ, instead of being in the range of 50, is estimated to be over 100. She's doing well in school. If you'll notice, her upper lip is kind of thin, but if you look around the room, there's a lot of us have thin upper lips. And if Sarah grows up and wants poochy lips, some plastic surgeon can provide that to her. That's not a great disability. But her heart, they said she's not going to require any surgery. We've never seen this. And they used some very special words. They said her heart developed like she was in her mother's uterus. And I was asked at that time, could I explain this? And I couldn't, because I hadn't seen what I've shared with you now. But you know why Sarah's heart developed like it was in the mother's uterus. These cells are those from the same source, migrated into the heart, just like a little 528-gram boy Trav in Cedar Rapids, Iowa, that had 18 premature diagnoses, had septal and cushion defects, not very well developed left diaphragm, and they thought he would have to have 3 surgeries. And I saw him at 27 months. He had caught up with all his peers, and they didn't have to do any surgery on him, and they said he developed like he was in his mother's uterus. We hadn't seen this, just like a 2-pounder out of Salt Lake City. Well, now we both know how this is possible through the cells that come from this source. Now, I wish that I could stand up here and put my thumb under my galluses and tell you how smart and brilliant and original all these things are, but in these 20 years of seemingly, because of my previous medical education, which did not include this, and knowledge, it seemed like it was climbing a black mountain of ignorance. And at different times when we found something, there was a tendency to want to go out on a ledge and shout to the world how great I am. But before I could get my mouth open, I would look on the mountain and find somebody else's name cut deeply in the stones. Deeply cut, like Hippocrates from 320 BC, ancient Greece, who left us translated from the Greek, let your food be your medicine and your medicine your food. Kind of hard to get ahead of him, isn't it? Isn't that humbling? In the Middle Ages, the most noted person from world history, from Switzerland, Paracelsus, said, all that man needs for health and healing has been provided by God in nature. The challenge of science is to find it. And every time that I show that assembly line that is in every one of your trillion cells, I think of the words of Sir Isaac Newton, who defined the scientific process, conditions, and gravity. He said, the purpose of science is to understand the mind of God. But in Texas, at the University of Texas, for over 40 years, there was a biochemist that wrote 12 books on nutrition. He says, the theme of my 12 books and of my life's work is in one sentence, the most important words that I will say to you tonight. And that is, quote, the human body heals itself, and nutrition provides the resources to accomplish the task. Tonight you have seen a new dimension in the wisdom of Dr. Williams' words. Thank you.